Thank you. Assalamu alaikum. Good morning. I'm Dr. Muhabat. I'll be taking you through the parasitology and mycology lectures. Since this is my first time of taking you through any session, I just want to tell you some things that are allowed during the lecture. If you have a question, most of my slides are numbered. It's either you note the number of the slide and ask your questions when I'm through, or you allow me to complete whatever it is I'm saying. And this is because, as you'll find out, everything I'm going to say is not on the slide. So if I'm trying to explain something and you cut in, I may lose my train of thoughts, okay? So allow me to complete whatever it is I want to say, then you can ask the question, or you can note the number of the slide and then you can ask the question later. Apart from that, my learning objectives are usually stated. These learning objectives are meant to guide your revision. My questions are going to be based on these learning objectives. Clear? I'm not going to ask you anything outside the learning objective. If after the discussion in the class and you have your private study and you still have questions, please feel free to send me an email, okay? So let's start. I'll be taking you through parasitology, as I said. This is a branch of microbiology that deals with parasites. So let me just go back to the first slide. You can see the cartoon and you can see the hippo complaining. I always give, and all you do is take. This represents a parasitic relationship. In any parasitic relationship, one host is always at the losing end or is harmed, while the other host is always benefiting, okay? So all we're going to be discussing for the next 3 lectures has to do with pathogens that have, evolved in such a way that they live on others, okay? So by the end of my discussion with you today, you should be able to discuss the different types of relationships that can exist between different organisms. You should be able to discuss important basic terms and concepts in parasitology. You notice that this is in red. It's very important. So at the end of today's discussion, if you cannot do this, you have not attended today's lecture. You should be able to discuss the classification of protozoa of medical importance. And when we talk about discussing these protozoan parasites, it's in terms of their life cycle, whatever that means, we're going to discuss it. And transmission, the epidemiology and diagnosis, prevention and control. So I'm not going to discuss the disease condition associated with this parasite. Is that clear? So let's start. If you have 2 organisms in an ecosystem, there's bound to be some form of interaction. So as I am in the class with you this morning, I'm trying to teach you something and you're trying to learn. So that's a form of interaction. So in an ecosystem, there's always that form of interaction between organisms, usually different organisms. So the term for that relationship is symbiosis. So symbiosis is the umbrella term. When we say symbiosis, we are referring to interdependence. How do we relate to one another? Under symbiosis, we have different types of relationships. So we have commensalism, we have mutualism, and parasitism, which is what we're going to discuss this morning. So what does those terms mean? In a commensalism, when you have species A and B, you find out that one species A, is benefiting from the relationship, while the second species is not affected, either negatively or positively. So the other host is just there. This is not so common in humans when we interact with other microbes. But in the environment, you may have this type of relationship in which at times some big trees will serve as support for other trees that are like crawlers. So these crawlers are able to carry out photosynthesis, but they don't have the height. So they only hang on to the big trees for support. The tree is not affected, the crawler plant is not causing any harm. Is that clear? So that's a form of commensalism. In mutualism, both agents that are involved in the relationship are benefiting. And a good example is our normal microbiota in the gut. We cannot make vitamin K, but the microbes in our gut can. And you know vitamin K is very important in the clotting process. So they produce vitamin K and we offer them shelter and food. So we are both benefiting. Is that clear? In parasitism, one organism receives all the benefits. The other organism is at a loss. And this is what this cartoon depicts. You can see he's finished his home food. He's now encroaching on the food of his other colleague. So that is parasitism in a nutshell. Now, as you must have realized from your bacteriology lectures, there are various types of microbes. And it only makes sense that we give them names so that if you are dealing with that particular isolate here in Saudi Arabia and you're on a conference call with colleagues on in other countries all over the world, you are all discussing the same organism. You are not, what you are discussing is not different from what your colleagues have in mind. So we tend to standardize our terms. So there are terms that we use in parasitology that you need to know. So when we say parasite, you already have an idea what that means. That's the organism that benefits from the relationship to the detriment of the other organism in the relationship, okay? So the parasite has what we call a host. The parasite cannot hang alone because it's causing damage to another organism. So that other organism is referred to as a host. Is that clear? So you have parasites, you have hosts. Clear? In terms of where the parasites are located on the host, we can classify them. So we have what we call ectoparasites and endoparasites. Ectoparasites are found on the outside of the host, so they don't invade, okay? And a very good example is the louse. We have head lice for plural, louse for one. So head lice is a good example. These are parasites that we can find on the head, and they are blood-sucking agents. Endoparasites live within the host, so they are found within the body or the tissue of the host cell. These include organisms such as tapeworm, malaria parasites, and microfilaria. For now, this may not make so much sense, but we are going to discuss these organisms. So for now, the take home is parasites may be outside on the host, living on the host, or they may be inside, living within the host. In terms of the biology We've discussed location now, we are clear. Now there's a biology to the parasite. So a parasite that requires a host all the time, it cannot exist without a host, is referred to as an obligate parasite, okay? A parasite that can switch between living on the host and living free in the environment is referred to as facultative parasite. And these are not new terms. I'm sure you've discussed in bacteriology, facultative anaerobes and strict anaerobes, right? So an obligate parasite may be regarded as a strict parasite. A host must always be present. A facultative anaerobe can switch. If there's a host, fine. If there's no host, the parasite can still exist. And you have examples such as Naegleria fowleri, Acanthamoeba, and Strongyloides decoralis. These are examples of parasites that are facultative. Then we have what we call accidental parasites. These occur when you have a parasite present in the non-usual host. I give you an example. You are all medical students registered here at Alfisa, right? Your records are here. If you sit for an exam, your results are going to be here. But let's say you now decide to go to King Khalid University, right? There's a College of Medicine, all right. And you decide to join the students there. You become an accidental medical student there because they don't have your records. If you sit for an exam there, that is, if you manage to get into the exam hall, your results cannot be collated there because they don't have your records. Exactly. So the moment you are there and you're attending lectures, you are an accidental medical student. So that's the same concept with the accidental parasite. So a good example is the dog tapeworm. It's usually supposed to be found in the dog. But at times, the eggs can find its way into our food or source of water, and we may be exposed to the parasite. But because we are not the usual host, it will not develop to maturity, but it can cause disease. Is that clear? So like I said, the host is the organism that is losing out in the interaction. Again, we can have different types of hosts, just like we talked about parasites that are facultative, they are obligate, accidental, and things like that. We talk of the definitive host and intermediate host. What makes a definitive host a definitive host? There are certain characteristics we look out for. We don't just look at the size and say, because this is a bigger host, this must be the definitive host. No. There are certain things we look out for. One is the biology of the parasite. For some parasites, as you're going to find out, you can have both sexual and asexual reproduction taking place in different hosts, okay? So the host in which you have sexual reproduction taking place is regarded as the definitive host because sexual reproduction is regarded as an advanced form of reproduction, more advanced than asexual. So the host in which you have sexual reproduction occurring is regarded as the definitive host. Again, as you're going to find out when we start discussing specific parasites, there are some parasites in which we are not sure or we can't identify whether sexual reproduction is occurring. But we can identify different forms of those parasites. The host in which you have the most developed form of the parasite, the most matured form, is regarded as the definitive host. So two things can define a definitive host: presence of sexual reproduction, or the most matured form of the parasite is found in that host. Are you following? The intermediate host is the host in which asexual reproduction occurs, or the host in which you have the juvenile form or the immature form of the parasite residing. So that is what defines a definitive or intermediate host. It is not the size of the host, as you'll find out. And I've given examples. With the malaria parasite, humans are the intermediate host, while the mosquito is the definitive host. We also have the host that we refer to as transport host. Now, this type of host does not take part in the biology of the parasite at all. All this type of host does is to physically move the parasite, the infected, from point A to point B. Okay, so that's why we say it's a transport host. A good example is houseflies. If you have an environment in which people defecate indiscriminately, and you have someone infected with, say, Entamoeba histolytica, the infected form are the cysts. These cysts are passed out in the feces. So if the flies perch on the feces in the environment and they find their way into the kitchen, and you have cooked food that is not covered, and the flies perch on the food, they can deposit the cyst from the feces that they have picked up on their body on the food. So all they have done is to mechanically or physically move the cyst from the feces in the environment right onto the cooked food in the kitchen. Are you following? Then we talk of the reservoir host. With the reservoir host, the parasite is present, but the patient or the host is not symptomatic. So the host will keep the parasite viable, that is, it keeps it alive, and in a form in which it can infect another host. That's why we say they're reservoir. So they keep that particular parasite viable and in a state in which they can transmit it to another host. Just like we have accidental parasites, we can have accidental hosts. So this happens when the particular host is not suited for that particular parasite, okay? And I think I gave the example of the dog tapeworm earlier. So it goes both ways. Humans are accidental hosts. The parasite is accidental in humans. Another term that we use is life cycle. Remember that with humans, life begins with fertilization of the ovum, then you're going to have the embryo, you're going to have the fetus, and then you're going to have the baby, right? That's our own life cycle in a way. For the lower forms, it's not as elaborate as what we have in humans or higher animals. So when you look at what happens from the infective stage through the development cycle, next to the next infective stage, the entire process is what we refer to as the life cycle of the parasite. So it means the sequence of developmental stages in the life of a parasite from one adult form of one generation to adults of the next generation. So here you have examples of a direct and an indirect life cycle. Now, whether it's indirect or direct, you'll notice from the diagrams that you have arrows that are leading from different forms from one host to another host. So it's a cycle. At each stage, you have different forms of the parasite showing maturation. Here you have a similar diagram showing development back to the infective stage. So it's a cycle. You have a complete cycle of events or sequence of events. Now, when we say life cycle, because we have different types of parasites, we can also subdivide the life cycle into indirect and direct. When we say direct, it means you require just one host for the entire sequence of development to occur. So everything occurs in one host. So that's direct. In indirect, you need two different hosts, okay? you need 2 different hosts to complete the life cycle of that parasite. And a very good example is the malaria parasite. We require both the mosquito and humans to make up the life cycle. So this is an example of an indirect life cycle, while this is an example of a direct life cycle. So why should you bother yourselves about parasitic infections? I'm sure most of you would think, well, do we need to know about parasites? The answer is yes. Parasitic infections are a global health problem. So even if you think in your own country parasites are not a problem, remember that now we have natural disaster, we have civil strife that has caused people to cross international boundaries as refugees. So if you're not conversant with parasites that are endemic in the areas where they're coming from, you may have this parasite introduced in your own country and you'll be caught unawares. So what are the factors that promote parasitic infections? As you're going to find out, most of these agents are associated with poverty in regions of the world where you have poor infrastructure, housing, health, and what have you. It's also, when you have parasitic infection, it may also give you an idea of the nutritional status of the environment. If you have high population density with inadequate housing, you may have people living on the streets and defecating in the environment. So that is going to enhance parasitic infection and transmission. In areas where you don't have potable water and people depend on open body of water, which may be contaminated with the parasitic agent, this will promote parasitic infection. Lack of personal hygiene, using the bathrooms, not washing your hand before you eat, shaking hands, not washing your hands after shaking hands because you just don't know where your friends' hands have been. So these are all factors that could lead to increased parasitic infection. And of course, lack of environmental hygiene and poor housing infrastructure and health facilities. So now let's calm down a little. We know the terms. We know the life cycle that can occur. Let's look at the different types of parasites that we can have. So when we talk of parasites, 2 broad classifications, the protozoa and the metazoa. What makes the protozoa? Because they are unicellular, just one cell. Okay, so it's based on the morphology of the parasite. Metazoa means multiple cells. So that's a basic classification. For the protozoa, which is what we're going to discuss today, you have those that we subdivide based on the presence of an organ of locomotion. So we talk of the sarcodina or the amoeba. They are the ones that move with the production of pseudopods, false feet. Okay, so they are motile and they can move by production of pseudopods. Then we talk of the sporozoa. The sporozoa do not actually have a recognized organ of locomotion. However, during development, they form an apical complex packed full of digestive enzymes. With the aid of these digestive enzymes, they are able to invade into the host cells. So they, although they don't have organs of locomotion in quotes, they are able to invade because of these digestive enzymes. At times, some books refer to them as apicomplexa. It's still the same group of organisms. You have the mastigophora. The mastigophora are the ones that move with the aid of the flagellum. Then we have the ciliata. These are the ones that have cilia, and the cilia are tiny ear-like structures that can beat and generate current that can make the organism to move. With the metazoa, we are not discussing them today, But just so that you know, these are made up of multiple cells. And they are the ones we refer to as helminths or the worms. They can be broadly divided into platyhelminths and nemathelminths. Platyhelminths are those that are flat dorsoventrally, like a tapeworm. Hence, they are usually referred to as tapeworms. Then you have the nemathelminths, which are the roundworms. They are the ones that look like the earthworm. With the platyhelminths, you can further subdivide them into the non-segmented trematodes. That is, they are not made up of units, they're just individual flattened organisms. And the class Cystoda, which are the segmented flatworms. So they have units that make up the entire organism. So how do parasites invade the host? We discussed now the routes of acquisition of different parasites. For some parasites, they can be inhaled. Remember I told you, poor environmental hygiene, poor housing infrastructure can promote parasitic infections. So when you have organisms such as Enterobius and Naegleria fowleri, Acanthamoeba, these organisms, Acanthamoeba and Naegleria, exist in the environment. When you have them dried up as cysts, air currents can disturb the cysts and you may inhale them. When they are inhaled, remember the mucociliary protective mechanism in the lungs, in the respiratory tract. So these cysts are entrapped in the mucus. And most of the time we don't spit it out, we tend to swallow it. So the cysts will find their way into the GI, where you can have development of the parasite. So that's one way of transmitting the infection. Then you have the fecal-oral or the ingestion of the infective form. As you can see, you have more parasites under this route of transmission. It makes sense. Most GI pathogens or parasites are acquired through the fecal-oral route. It makes sense, right? So you have a lot of organisms. Now, before you ask, do you need to know everything? Maybe one or two. But you need to recognize these routes of transmissions, okay? You can have parasites through direct sexual contact, okay? We are going to discuss Entamoeba and Giardia. You may wonder, these are GI pathogens. Why are we saying they can be transmitted sexually? It depends on the patient's sexual orientation. There are men who have sex with men, so they can end up infected with this parasite because of their sexual orientation, okay? So it's not the usual route, but it is a known route of transmission in a subset of patients. Is that clear? Trichomonas is a known organism that is sexually transmitted, okay? You can also have transmission through the penetration of intact skin. Some parasites are so vicious, if they come into contact with the skin, they can actually find their way through the skin. So you can have contact and penetration of the skin. You also have those that are vector-borne. Vector in the sense that you have another organism that feeds on the host. So in the process of feeding, the parasite is introduced. So you have vector-borne parasites. You can also have parasites transmitted through contact and penetration of the eye, not just the skin. So some parasites can actually enter through the eyes. So we've discussed how parasites may be acquired, parasitic infections may be acquired. What about the tissue in the host that is affected? Now, this is just my own classification to help you with your reading. You're not likely to find this in your textbook, okay? So with the protozoan parasites, remember they are unicellular. And we have different parasites. We have those that cone down on the gastrointestinal tract. So we have those that cone down on the gastrointestinal tract. So you find the adult parasite in the GI, and you have examples listed there. You have those that go for tissues other than the GI. So you can have them in the brain. You can have them affecting the cornea, the eyes, or you can have them affecting the genital tract. So those ones are found in the tissues. And you have parasites in the blood. Okay, and you have examples here. Like I said, this is not in your textbook. It's just to help you. But when we start discussing the life cycle, it gives you an idea why they are transmitted the way they are. Okay. So we start with the GI protozoan parasites. Starting with Giardia lamblia. We're going to discuss this protozoan parasite in terms of its life cycle. The life cycle is made up of two stages. These are the two stages, the cyst and the actively metabolizing trophozoite, which is pear-shaped. Now, the cyst is regarded as the infective form. This is the form of the parasite that can establish infection in a susceptible host. If you don't come into contact with the cyst, you don't get an infection. Is that clear? The trophozoite is what emerges from the cyst. This is the actual form of the parasite that causes the disease, giardiasis. Is that clear? So I want you to get that difference. When we say infective form, that is the form of the parasite that will establish infection in a susceptible host. Are you following? The trophozoite is that stage of the parasite that is actually responsible for the clinical disease, right? So the life cycle consists of two stages, the trophozoite and the cyst. The trophozoite is pear-shaped with two nuclei, 4 pairs of flagella, so it is a motile organism, and a suction disc with which it attaches to the intestinal wall. Now, this is very important. This particular parasite is not invasive. It's only going to bind onto the enterocytes using the suction cup. Clear? It's not invasive, yes.
Does this make it extraparasitic?
What do you think? Where is it found? The GI. Oh, is that internal or external? Internal. So this is an endoparasite. Okay? If it is found on the skin, that will make it ectoparasite. If it is found on the scalp, that will make it ectoparasite, but it is internalized. Clear? Good. The oval cyst is thick-walled. This is because the cyst needs to survive in the environment. If the cyst is not thick-walled, it's going to be destroyed. So having a thick wall makes it resistant to drying. So the cyst has a thick wall with 4 nuclei, as you can see in the diagram, and several internal fibers, which will eventually grow up, develop into the flagella. Each cyst gives rise to 2 trophozoites during excistation, that is, during the emergence of the trophozoite from the cyst. So you can see 1 cyst will give rise to 2 organisms. So if you ingest 10 cysts, what will happen? Exactly. So how do we end up with infection? So we're going to discuss transmission, life cycle, and pathogenesis. In terms of transmission, we've already said it that the cyst is the infective form. And this is a GI pathogen. So how can the cyst get into the GI? Through contaminated food, contaminated water, contaminated fingers, If you have food handlers who are carriers and they're serving you food, they can contaminate the food, okay? So when we discuss life cycle, we start from what happens with the definitive host. In this case, because we have a direct life cycle, we can't talk of definitive and intermediate host. So we start from the host. Now, an infected host will pass out both the cyst and the trophozoite in stools. But because the trophozoites are fragile, they don't have a thick wall, they will die in the environment. The cyst will persist. So the cysts eventually find their way into the food chain. And if we are not careful, for example, we eat raw vegetables that are not well washed with potable water, or we drink contaminated water, we're going to ingest the cyst, okay? And then the cyst gets into the GI. The digestive enzymes will dissolve the thick coat, and then the trophozoites will emerge. And then an infective process occurs. In terms of pathogenesis, the trophozoite causes inflammation of the duodenal mucosa, leading to malabsorption of protein and fat. That's the main pathology of this parasite. Remember, I said it's not invasive. So while they are stuck onto the enterocytes, they are living. So they are producing their own waste, which the body will pick up as not normal. So you're going to have some form of inflammatory response to their presence in the GI. Apart from the fact that you have the inflammatory response going on, when they bind onto the enterocytes, remember that the enterocytes have microvilli. They're going to cause disruption of the microvilli. Remember that the microvilli give us a wider surface area for absorption. So when they destroy these microvilli, then we're going to have a reduced surface for absorption. So we have malabsorption of protein and fat. Because apart from the fact that the microvilli give us large surface area, they also have enzyme in the brush borders, right? Yes, what's your question?
Does this act like spore?
We don't use the term spore, but it's a survival stage, similar to what the spore does, but they are not spores. Okay? Yes.
Does the cyst multiply or just the number of weeks?
No, the cyst does not multiply. The cyst is metabolically inactive. Any other questions? Now, in terms of epidemiology, this parasite is found worldwide. Approximately half of those infected are asymptomatic carriers. This is part of the reason why it's difficult to eradicate this parasite. If you're not showing symptoms, you're not likely to present to the clinic for treatment. For people who have immunoglobulin A deficiency, they are at a higher risk of coming down with this parasitic infection due to this parasitic agent. Remember that immunoglobulin A is on the mucosa, so it's supposed to protect us. So where you have people with immunoglobulin A deficiency, they are unduly susceptible. Outbreaks related to contaminated water supplies have been known to occur. So there's that common source of exposure. One thing you should note is that chlorination does not kill the cyst. So because you have potable water from the tap at home does not mean if you have a contaminated source that you are free of the cyst. The amount of chlorine, residual chlorine, in potable water that gets to the home does not destroy the cyst. But if you have a filter attached to the tap, then you can say that you're free of the cyst. In those who go camping, hikers who drink untreated stream water, they are at a greater risk of infection. This is because unlike Entamoeba histolytica, Giardia lamblia can also infect other vertebrates apart from humans. So wild animals such as bears may be infected. These are animals that can contaminate open body of water. So if you go hiking and you think the stream water is clean, just because it appears clear, you may be at risk of ingesting the cyst. Giardiasis is common in male homosexuals as a result of oral anal contact. Incidence is high among children in daycare centers and patients in mental hospitals. Now, the reason why it's higher in children in daycare centers is that you may have shared toys. So if you have a baby caregiver who is infected, asymptomatic, he or she is shedding the cyst and is not very good with personal hygiene, he or she may contaminate the toys. And you know, children, they experiment through the mouth. They always stick things in their mouth. So the risk of picking up the cyst from contaminated shared items is higher. In patients in mental hospital, these are people that may have poor personal hygiene. So it's easier for this parasite to, be transmitted in such settings. Like I said, we are not going to discuss the disease condition. It causes diarrhea. You can know that, okay? And we describe it as fat, bulky, foul odor, okay? So the diarrhea is non-bloody. How can you make a diagnosis if you suspect a patient has giardiasis? Because there are different organisms that could cause diarrhea. Stool sample will be a good one to start with. Why do you think stool sample is a good? Very good. It's found in the GI. So it makes sense that whatever happens will reflect in the stool, right? We said it's not invasive, so we don't think we find anything in the blood, say for example, right? But the problem is the cysts are not shed all the time. So you don't know when the patient is going to shed the cyst in the stool. As a result, we send at least a minimum of 3 stool samples on different days so that we can increase the probability of getting the cyst or the trophozoite in the stool sample. Now, we already described the morphology of the cyst and the trophozoite. So this is what we look out for in the stool sample. If the stool sample gets to the lab on time, it's possible to see the trophozoites before they disintegrate. However, if we don't get the stool sample on time, we can still look out for the cyst. Remember that the cyst has a thick wall, so they are resistant to drying. You can also send in a duodenal aspirate. Remember that these trophozoites are going to emerge in the duodenum. So you can perform the string test or the entero test, in which the patient will swallow a capsule. The contents of the duodenum will be collected in the capsule. You retrieve it and you look at it under the microscope. You can biopsy. Of course, this is not coming to microbiology lab. This will go to the histology lab. We can also perform fecal antigen detection tests. There are different test kits that can do this. Because this is not a human cell, there are antigens associated with it that the body will recognize and the body will respond to. So there are different approaches: immunoelectrophoresis, enzyme immunoassay, immunochromatography. And of course, you can use molecular assays. These are nucleic acid amplification tests. So things like DNA probes and polymerase chain reaction may help. So we move on to the next parasite, Entamoeba histolytica. Again, this is an example of a GI pathogen. So where do you expect to find the organism? In the GI, right? The life cycle, just like what we have with Giardia, has 2 stages. Here you have the cyst and the trophozoite. The trophozoite is found within the intestine, and you can also have extraintestinal lesions. This is a marked difference from Giardia. Giardia is non-invasive. Entamoeba is invasive. As a result, you can have a GI pathology, or you can have an extraintestinal pathology. Is that clear? The cyst predominates in non-diarrheal stool, that is formed stool. And this makes sense. The trophozoite is very fragile. So if the stool is formed, they die off readily. But in the loose diarrheal stool, it's possible to find the cyst. The cysts are not resistant to boiling, so boiling of water will take care of the cyst. They can also be removed by filtration because they are resistant to chlorination, just like what we had with Giardia. Now, since the cysts are forms of the parasite that we can use to make a diagnosis, we have to look at features that makes them different, for example, from that of Giardia, because we said cyst, cyst, cyst. So how do I know this cyst is for Entamoeba histolytica? The cyst, when it is matured, has what we call 4 nuclei. So these are the 4 nuclei, 1, 2, 3, 4. It is an important diagnostic criterion. So we actually look out for the cyst with 4 nuclei to make a diagnosis. If the cyst is not matured and we have less than 4 nuclei, it's not so easy to make a diagnosis. Clear? Now, when the trophozoite emerges, the amoeba has 4 nuclei, and then it divides to form 8 trophozoites. The mature trophozoite has a single nucleus with an even lining of the peripheral chromatin. So what does this mean? When the cyst gives rise to the trophozoite, we look out for some morphological forms to differentiate the trophozoite of Entamoeba histolytica, which is pathological, from those of non-pathological Entamoeba. Okay? With Entamoeba histolytica, if you look at the nucleus, this is it. What we are saying here is that the chromatin pattern is smooth and evenly distributed as shown in this diagram. Antibodies are formed against trophozoite antigen in invasive disease, but they are not protective. Previous infection does not prevent reinfection, okay? The antibodies that are produced are useful for serological diagnosis. So what is the life cycle like? This is a direct life cycle, just like what we had with Giardia, because we need just one host. And the life cycle starts when you have an infected host, passing out the trophozoites and the cyst in stool. Remember that the trophozoites are very fragile, so they are going to die off in the environment, but the cysts are hardy, they will survive. So if the cysts find their way into the food chain and they are ingested, then the life cycle continues. Now, one thing you should note is that with Entamoeba histolytica, unlike Giardia, we don't have animal reservoir. Only humans are known to transmit this parasite. So hikers are not at risk of coming down with Entamoeba histolytica unless you have human feces contaminating that stream. Okay? Another thing that you should also note is that you can have extra-intestinal infection, and this is represented by the arrows that are leaving the GI. So you can have the parasite finding its way into the bloodstream, and from the bloodstream, you may have involvement of the liver, you may have involvement of the lungs, or you may have involvement of the brain. So you can have extra-intestinal conditions with Entamoeba histolytica. That does not occur with Giardia. Clear? The trophozoites, because they are invasive, they can invade the colonic epithelium and secrete enzymes that cause local necrosis. So if you take a biopsy, you have what we call the flask-shaped ulcer. So I've put here a picture of the conical flask to give you an idea of why it is so named. If you look at this histological section, you have the narrow neck through which the organism invaded, and then due to the hydrolytic enzymes, you can see the extensive necrosis below the surface of the mucus. So this is why we say they are flask-shaped ulcers. Just like with Giardia, infection occurs worldwide, although it occurs frequently in tropical countries with poor sanitation. Tropical countries, because the environment is warm and humid, so the cyst can persist for a longer time in the environment. Infection is also common in men who have sex with men. Now, how do you make a diagnosis? If the patient has a GI infection, again, the stool sample is your best sample to send to the lab. Again, you have to send a minimum of 3 on different occasions or different days. And that's because the cysts are shed intermittently. So we look out for the characteristic cysts and the trophozoites if they are present, okay? You can perform enzyme immunoassay using fecal antigens because there are some antigens, we said it, that are associated with the parasites. So we can screen the stool sample for fecal antigen. You also have molecular assays, nucleic acid amplification tests like polymerase chain reaction and DNA probes. I'll quickly go through this, but you're still going to have this in the lab. But when you go to the lab, I just want you to now apply what I'm going to discuss with you to the slides that you're going to be provided with. Clear? Remember I said Entamoeba histolytica is pathogenic. It's always pathogenic. But we also have non-pathogenic Entamoeba strains that may be present in a stool sample. So if you have a stool sample, there are certain things we look out for to make up our mind whether you have the pathogenic strain or the non-pathogenic strain. The first thing is to look at the nucleus of the trophozoite. Remember, I told you earlier that the chromatin dots are evenly dispersed at the periphery of the nucleus. So Entamoeba histolytica nucleus has a small central nucleolus, which is what you have here. This is the nucleolus, and fine chromatin granules along the border of the membrane, which is what you have here. It is evenly distributed. So we look out for these under the microscope. As you can see here, you have a micrograph showing the trophozoites of Entamoeba. You can see the location of the nucleolus, and you can see the margin of the nucleus, that it is evenly dispersed. Clear? If you look at the non-pathogenic Entamoeba, for example, Entamoeba coli, the nucleolus is eccentrically located. It's not found in the center. Apart from that, the chromatin particles, as you can see, they are in clumps. They are not evenly dispersed. So if we observe this, we know that we are dealing with a non-pathogenic strain. And here you have a micrograph showing you a non-pathogenic trophozoite. As you can see, it's not evenly distributed. You can see that here, it's like you don't have any chromatin deposition. Here you have, okay? Another thing we look out for are the cyst size and the number of nuclei. Here you have the cyst of Entamoeba histolytica. The matured cyst has 4 nuclei. With the non-pathogenic Entamoeba coli, you have more than 4, okay? So if we see this, we know this is non-pathogenic. One thing you should also note is that there is a non-pathogenic strain of Entamoeba, Entamoeba dispar, that is so similar to that of the cyst, is so similar to that of Entamoeba histolytica in that it has 4 nuclei. So when we see 4 nuclei at times, we report as cyst of Entamoeba histolytica / Entamoeba dispersing. The only way we can differentiate between these 2 species is to perform a molecular assay. Clear? Now, the third thing we look out for, particularly if we have trophozoites in the stool sample, is to look at the directionality of the trophozoite. With pathogenic Entamoeba, they move in one direction only, and they can actually move up the slide when you mount them. But with the non-pathogenic entamoeba, it's like Brownian motion. So they are going, okay, just 2 minutes and I'm done. They're going in all the directions, okay? So how can you prevent? Hand washing, good personal hygiene, boiling, filtering your water, good waste disposal, washing raw fruits with potable water, making sure that cooked food are covered so that flies cannot perch on it and things like that. And have a nice day. You're welcome. I'm sorry.